Hello, today we're going to talk about successful case of biomonitoring in an aquatic ecosystem. This is an assignment from our topic of aquatic biotechnology and our teacher is Lourdes Diaz Tenorio and we are Team Aguachiles. We hope you enjoy our presentation. Our study took place in Sao Paulo and Rio Grande, Brazil. This is a huge country that has a lot of terrain covered with a lot of forest. Most of these forests are rainy, so the conditions over there are very humid. The main body of water that crosses a huge portion of the country and also out of the country is the Rio Amazonas. But now let's focus in the mainland that this investigation took place on. In the graphic, we can see some abiotic factors, like temperature and the quantity of rain that falls during the year. The columns represent the rain accumulated during specific periods of time. We can identify that we have a very opposite kind of climate compared to us. Like in December is cold for us, but for them it's a warm climate. And also the rain seasons change with it. Analyzing the graphic, we can see that from December till June, the rains are very present with a lot of millimeters of water accumulated, and from June to September, it's a little less frequent. Now, focusing in the lines following our legend, we can see that the solid lines represent the average of temperature, which is mostly warm and a little bit cold. The temperatures can be between 10 to 23 degrees Celsius, making it a good climate. At least for us, that we have more kinda 40 degrees, something like that, which is a very high temperature. The temperatures may go as low as 3 degrees Celsius, which is marked in the graphic. This is marked with the dashed lines. And the hottest one, which are the dot lines, it can be seen as 27 degrees Celsius, which is a paradise for us. This data was extracted from a work of Marchioro and company in 2003, meaning that the data won't be the same as today, but it can be used to give us an idea about its abiotic factors on that time. This table was extracted from an article written by Mantellato and Company in 2003. The data is from 1995, so the numbers may be different if we start to analyze the land with the same methods, due to the changes occurring with the time. We can see the salinity factor being kind of high. This is because the place where it was analyzed is a bay. This bay is called Ubatuba Bay, and it's located at the coast of Brazil at Sao Paulo. In only 1 year, the salt levels were stable because it goes up and down with some months, meaning that the ocean maybe took some quantity of desalinity or something like that. One of the most noticeable changes is the amount of organic matter that was a lot in October. Maybe to an error or a disaster, or maybe one possibility can be because the people were going to the beaches and they were having some fun and the organic matter went up because it's when the warm climate starts to occur. The next change that was noticeable was during July, where the concentration started to go up again. This is a very alarming data because it means that the organic matter is going up, and we have to find a solution to reduce the concentration of it. The main problem that this land suffers are the three main types of pollution that we all experience around the world. These are the industrial, urban, and agricultural pollution. The industrial pollution is a big thing in Brazil because some studies in 1999 identified huge concentration of heavy metals and strange organic matter. Its origin was traced to a near fabric, but due to the law enforcement being kind of weak, and that sometimes it's cheaper to pay the fine instead of installing a purifying chamber inside the fabric, they decide to pour all of its waste to the ocean or a near river. Now, talking about the urban pollution is due to extreme poverty and a huge population. There is a lot of people that instead of throwing the trash into a trash can or a designed place for it, they only throw it to the streets and the wind takes it to the ocean or a near water body. This trash includes a lot of oils, medicines, plastic, and the food, which can be considered as a major contributor of the organic matter being so high. We also have to consider its beaches and the tourism it used to receive before the pandemic started. The agricultural pollution in Brazil has always been a problem. Since 2008, Brazil has been the largest consumer of these products, which produce the pesticides, the herbicides, and all those kind of products. And since January 1st, 2019, under the administration of Bolsonaro, the government has discovered that there are around 945 new pesticides that has been used in a national way, which 311 are prohibited components in the European Union due to a high degree of toxicity that these have. The use of these products is causing diseases to human life, like cancer, poisoning, seizures, damage to the nervous system, among other diseases.
From the bioindicator in Brazil, the 62.6% of bioindicators were carried out exclusively with fish. On the table that is in this page, you can see the organism, the number of the study, and the percent. For example, the fish, the number of studies is 62 and 62.6%. From the bivalves, the number of the studies is 17 and the percent is 70.2. From the plants, the number of the studies is 6 and the percent is 6.1. Other organisms, the number of the study is 11 and the percent is 11.1. And organisms with more one type of bioindicator, the number of studies is 3 and the percent is 3%, from a total to 99 number of studies and a percent with 100%. In this page you can see many types of fish that you can extract bioindicators. For example, the fish Astanex river, the fish Deuteron iweb, the fish Hollandischit multifaciens, the fish Hypes bricon grami, the fish Hypos bricon bolungeri, the fish Maimajonits microlepsis, the fish Oligocerus epsetus, and for the end, the fish Cheiricium lanei. In this page you can see an example from a bivalve. From this picture is a bivalve that the name is Guayunella clenchi, and you can see the different types of form from this bivalve. The commercial and the ecological importance from the bioindicators. The bioindicators have much importance because for they you can do many studies that these studies can be developed and we can have the information and the benefits from the health and the biotechnology the benefits from the health and the benefits from the biotechnology for the bioindicators. And from the biomarkers in these tables you can see different type of biomarkers that you can extract or study in Brazil. For example, the micronucleus test, the glutathione S transferase, the catalase, the histopathological analysis, the comet assays, the morphometric index, the acetylcholinesterase activity, the lipoperoxidation analysis, the metallothionein, the superoxide dismutase, and others. The number of the studies in total is 52, and you can see the percent. The percent is 52.5, and for the next pages you can see the importance and how they worked on the studies. The types of the study that you can use the biomarkers. The studies using biomarkers for the evaluation of aquatic ecosystems can present different approaches. They can be field studies, they can be laboratory studies, and with caged organisms. Considering studies carried out on Brazil, the 68% were field studies, the 19% were laboratory studies, the 10% were with cages organisms, and the 3% is a study combining laboratory and the cage animals.
Micronucleus test is a one of biomarker. This biomarker is a test used in toxicological screening for potential genotoxic components. The assay is now recognized as one of the most successful and reliable assays for genotoxic carcinogens that act by causing genetic damage and is recommended by the OECD winning line for the testing of chemicals, and there are two major versions of this test, one in vivo and the other in vitro. Glutathione S transferase can constitute up to 10% of cytosolic protein in some mammalian organs. GSTs catalyze the conjugation of GSH via a sulfhydryl group to electrophilic centers on a wide variety of substances in order to make the components more water soluble, and this activity detoxifies endogenous components such as peroxidized lipids and enables the breakdown of xenobiotics. Catalase is a common enzyme found in nearly all living organisms exposed to oxygen, such as bacteria, plants, and animals, which catalyze the decomposition of hydrogen peroxide to water and oxygen. It's a very important enzyme in protecting the cell from oxidative damage by reactive oxygen species. Topological analysis is the gold standard for tissue examination, either for research or diagnostic purposes, for both qualitative and quantitative measures. It's used to assess the inflammation, or healing stage and to monitor the presence and distribution of degradation products that dissolve into the surrounding tissue. Different staining is used to identify certain structures, cells, tissues, or even metals components. Comet assay is an uncomplicated and sensitive technique for the detection of DNA damage at the level of the individual eukaryotic cell. Is a simple method for measuring deoxyribonucleic acid DNA stand breaks in eukaryotic cells. Morphometric index refers to the qualitative analysis of from a concept that encompasses size and shape. Morphometric analyses are commonly performed on organisms and are essential in analyzing their fossil records. The impact of mutation on shape, developmental changes in from covariances between ecological factors and shape, as well for estimating quantitative genetic parameters of shape.
Acetylcholinesterase is a human enzyme of the cholinesterase family found in nervous tissues and red blood cells, whose main function is to hydrolyze the neurotransmitter acetylcholine. And in red blood cells, it constitutes a cellular antigen called Ucii. Methylthionine is a family of cysteine-rich, low molecular weight proteins. They are localized up to the membrane of the Golgi apparatus. The induction of the synthesis of MTS in many marine species by contaminating metals has led to the proposal of the use of these proteins as a potential specific biomarkers for exposure to metals and toxicity in aquatic biomonitoring, provided tactic factors, natural and physiological, are taken into account. Superoxide dismutase is an enzyme, formerly hemocoprine and erythrocoprine, catalyzes the dismutation of superoxide into oxygen and hydrogen peroxide. Um, it is used in cosmetic products to reduce free radical damage into skin, for example, to reduce fibrosis that occurs as a result in radiotherapy. Biomonitoring or biological monitoring can be defined as the systematic use of biological response to evaluate changes in the environment. In this context, biomarkers biomarkers are increasingly worldwide recognized tools for the assessment of pollution impacts, and some are already incorporated in environmental monitoring programs in other countries, although their systematic and large-scale application is rare. Fish can be considered suitable organisms for biomonitoring studies as they are sensible to change in the aquatic environment. Their biological response change even at low levels of pollution. Fish can be found virtually everywhere in the aquatic environment, and they play a major ecological role in aquatic food webs as carriers of energy from lower to higher topic levels. Thus, genetic, biochemical, and morphological response represents useful biomarkers in environmental biomonitoring. In addition, the high percentage of studies carried out using fish as bioindicator can be also explained by the simple fish samplage techniques.
That data analysis points towards a limited use of this approach in the country because it is restricted to these two regions that were mentioned before. A great variety of organisms used as bioindicators and a different sampling patterns. That different sampling patterns can be described as a poor practice inside the investigation area. In order to have a more homogeneous data management, it's preferred to have a more static way on taking samplings. This way, the data will be more consistent. In the table, we can see a disproportionately sampling pattern, being the lowest 9.1 and the highest 31.3%. A great number of studies were conducted using fish as bioindicator organisms, but the water physicochemical analyses are still poor and fail in providing information to establish relations between the biomarker responses and the contaminants. The physicochemical analysis can be prioritized more in the future. Using only indicators is a good way to tell if there's some kind of contamination, but the analysis of these parameters allow us to know in a more quantitatively way to know how bad the situation is in order to take some actions.